Welcome to the video lecture for Unit 3, Module 6.3. In this one, we're going to elaborate where we left off in the last module and discuss more specifically about T cells, B cells, and immunity. As always, here are the learning objectives for this module. Use these when studying both for the unit quiz as well, or for the module quiz as well as the unit exam. And let's go ahead and get started. So the role of T cells in the immune response is varied and important in resisting pathogens. We've already noted four major types of T cells. You have cytotoxic T cells, which are responsible for cell-mediated immunity. These cells enter peripheral tissues and directly attack antigens physically and chemically. Memory T cells respond to antigens that they have already encountered by cloning more lymphocytes to ward off the invader. Helper T cells stimulate the responses of both T cells and B cells. Helper T cells are absolutely vital to the immune response because they must activate B cells before the B cells can produce antibodies. The reduction in helper T cell population that occurs in diseases like AIDS is largely responsible for the loss of immunity. And lastly, suppressor T cells inhibit T cells and B cell activities and moderate the immune response. Before an immune response can begin, T cells must be activated by exposure to an antigen. This activation seldom occurs through direct interaction between the T cell and the antigen. Foreign substances or pathogens entering a tissue commonly fail to stimulate an immediate immune response. For T cells to recognize an antigen, the antigen must be bound to glycoproteins in the plasma membranes of another cell. Recall that glycoproteins are integral membrane components. Antigen presentation occurs when an antigen-glycoprotein combination capable of activating T cells appears in plasma membranes. The structure of these glycoproteins is genetically determined. The genes controlling their synthesis are located along one portion of chromosome 6 in a region called the Major Histocompatibility Complex, or MHC. These membrane glycoproteins are called MHC proteins, or human leukocyte antigens. The amino acid sequence and the shapes of the MHC proteins differ among individuals. Each MHC molecule has a distinct three-dimensional shape with a relatively narrow central groove. An antigen that fits into this groove can be held in position by hydrogen bonding. Two major classes of MHC proteins are known. We have class 1 and class 2. An antigen bound into a class 1 MHC protein acts like a red flag that tells the immune system, hey, I'm an abnormal cell, kill me. An antigen bound to a class 2 MHC protein tells the immune system, hey, this antigen is dangerous, get rid of it. Class 1 MHC proteins are in the plasma membranes of all nucleated cells. These proteins are continuously synthesized and exported to the plasma membranes and vesicles created by the Golgi apparatus. As they form, class 1 proteins pick up small peptides from the surrounding cytoplasm and carry them to the cell surface. If the cell is healthy and the peptides are normal, T cells will ignore them. If the cytoplasm contains abnormal or non-self peptides or viral proteins, they soon appear in the plasma membrane, and T cells will recognize them and will be activated. Ultimately, their activation leads to the destruction of abnormal cells. This is the primary reason that donated organs are commonly rejected by the recipient. Despite preliminary cross-matching testing, the recipient's T cells recognize the transplanted tissues as foreign. Class 2 MHC proteins are present only in the plasma membranes of antigen-presenting cells and lymphocytes. Antigen-presenting cells, or APCs, are specialized cells responsible for activating T cell defenses against foreign invaders, including bacteria and foreign proteins. Antigen-presenting cells include all phagocytic cells of the monocyte macrophage group discussed in the earlier modules, including 1. Free and fixed macrophages in connective tissues, 2. The Kupffer cells of the liver, and 3. The microglia of the central nervous system, the dendritic or the Langerhans cells of the skin, and the dendritic cells of the lymph nodes and spleen are the APCs that are not phagocytic. Phagocytic APCs engulf and break down pathogens or foreign antigens. This antigen processing creates fragments of the antigen, which are then bound to class 2 MHC proteins and inserted into the plasma membrane. Class 2 MHC proteins appear in the plasma membrane only when the cell is processing antigens. Exposure to an APC membrane containing a processed antigen can stimulate appropriate T cells. The dendritic cells remove antigenic materials from their surroundings by pinocytosis rather than phagocytosis. However, their plasma membranes still present in antigens bound to class 2 MHC proteins. More important is that, unlike macrophages, they do not migrate very far from an infection site. Dendritic cells travel with their foreign antigens to lymphocyte-packed lymph nodes. So how do T cells recognize antigens? Inactive T cells have receptors that bind either class 1 or class 2 MHC proteins. These receptors also have binding sites for specific target antigens. If an MHC protein contains any antigen other than the specific target of a particular kind of T cell, the T cell remains inactive. If the MHC protein contains the antigen that the T cell is programmed to detect, binding will occur. This process is called antigen recognition because the T cell recognizes that it has been found by an appropriate target. Some T cells can recognize antigens bound to class 1 MHC proteins, whereas others can recognize antigens bound to class 2 MHC proteins. Whether a T cell responds to antigens held by one class or the other depends on the type of protein in the T cell's own plasma membrane. The membrane proteins involved are members of a larger group of proteins called CD, or clusters of differentiation markers. Lymphocytes, macrophages, and other related cells have CD markers. More than 70 types of CD markers exist, and each type is designated by a number. All T cells have a CD3 receptor complex in their plasma membranes, and this complex ultimately activates the T cell. Either of two other CD markers may be found bound to the CD3 receptor complex, and these two CD markers are especially important in specific groups of T cells. One, the CD8 markers are found on cytotoxic T cells and suppressor T cells, which together are often called CD8 T cells or CD8 plus T cells. CD8 T cells respond to antigen presented by class 1 MHC proteins. And two, CD4 markers are found on helper T cells, or often called CD4 T cells or CD4 plus T cells. CD4 T cells respond to antigen presented by class 2 MHC proteins. T cells are not usually activated upon their first encounter with the antigen. Antigen recognition simply prepares the cell for activation. In order to proceed from recognition to activation, a T cell must also bind to the stimulating cell at a second site. This vital secondary binding process, called co-stimulation, essentially confirms the initial activation signal. Co-stimulation is like the safety on a gun. It helps prevent the T cells from mistakenly attacking normal or self tissues. If a cell displays an unusual antigen but does not display the I am an active phagocyte or I am infected signal, the T cell activation will not occur. Co-stimulation is important only in determining whether a T cell will become activated. Once activation has occurred, the safety is off and the T cell will attack any cells that carry the target antigens. Two different types of CD8 T cells are activated by exposure to antigens bound on class 1 MHC proteins. One type responds quickly, giving rise to large numbers of cytotoxic T cells and memory T cells. The other type responds more slowly and produces relatively small numbers of suppressor T cells. Cytotoxic T cells seek out and destroy abnormal and infected cells. They are highly mobile cells that roam throughout intertissues. When a cytotoxic T cell encounters its target antigen bound to a class 1 MHC proteins, it immediately destroys the target cell. The T cell may 1. Release perforin to destroy the target cell's plasma membrane, 2. Secrete a poisonous lymphotoxin to kill the target cell, or 3. Activate genes in the target cell's nucleus that tell the cell to die. The entire sequence of events from the appearance of the antigen in a tissue to cell destruction by cytotoxic T cells takes a significant amount of time. After the first exposure to an antigen, 2 days or more may pass before the concentration of cytotoxic T cells reaches effective levels at the site of an injury or infection. Over this period, the damage or infection may spread, making it more difficult to control. The same cell divisions that produce cytotoxic T cells also produce thousands of memory T cells. These cells do not differentiate further in the first time the antigen triggers an immune response. However, if the same antigen appears a second time, memory T cells immediately differentiate into cytotoxic T cells. They produce a prompt, effective cellular response that can overwhelm the invader before it comes well established in the tissues. Suppressor T cells suppress the response of other T cells and B cells by secreting inhibitory cytokines called suppression factors. Suppression does not take place right away, because activation takes much longer for suppressor T cells than other types of T cells. In addition, upon activation, most of the CD8 T cells in the bloodstream produce cytotoxic T cells rather than suppressor T cells. As a result, suppressor T cells act after the initial immune response. In effect, these cells limit the degree of immune system activation from a single stimulus. Upon activation, helper T cells with CD4 markers undergo a series of divisions that produce both active helper T cells and memory helper T cells, also called memory TH cells. The memory helper TH cells remain in reserve. The active helper T cells secrete a variety of cytokines that coordinate specific and nonspecific defenses and stimulate cell-mediated and antibody-mediated immunities. These cytokines do the following. They, 1, stimulate the T cell divisions that produce memory helper T cells and speed the maturation of cytotoxic T cells. 2, they enhance nonspecific defenses by attracting macrophages to the affected area, preventing their departure, and stimulating their phagocytic activity and effectiveness. 3, attract and stimulate the activity of cytotoxic T cells, providing another means of destroying abnormal cells and pathogens. And 4, they promote the activation of B cells, leading ultimately to the antibody production. The plasma membranes of infected or otherwise abnormal cells triggers an immune response when CD8 T cells recognize antigens bound to class I MHC proteins. Extracellular pathogens or foreign proteins trigger an immune response when CD4 T cells recognize antigens displayed by class II MHC proteins. As we noted earlier, the body has millions of B cell populations. Each B cell carries its own particular type of antibody molecules in its plasma membrane. If corresponding antigens appear in the interstitial fluid, they interact with these superficial antibodies. When binding occurs, the B cell prepares to undergo activation. This preparatory process is called sensitization. Because B cells migrate throughout the body, pausing briefly in one lymphoid tissue or another, sensitization typically takes place within the lymph node of the nearest site of infection or injury. Recall that B cell plasma membranes contain class II MHC proteins. During sensitization, antigens are brought into the cell by endocytosis. The antigens subsequently appear on the surface of the B cell, bound to class II MHC proteins. The sensitized B cell is then on standby, but generally does not undergo activation unless it receives the OK from a helper T cell. The need for activation by helper T cells helps prevent inappropriate activation, the same way that a co-stimulation acts as a safety in cell-mediated immunity. So what happens when a sensitized B cell meets a helper T cell that has already been activated by an antigen presentation? The helper T cell will bind to the B cell's MHC complex. It recognizes the antigen and then begins secreting cytokines that promote B cell activation. After activation of the B cell, these cytokines stimulate the B cell division, speed plasma cell formation, and enhance antibody production. The activated B cell typically divides several times, producing daughter cells to differentiate into plasma cells and memory B cells. The plasma cells begin synthesizing and secreting large quantities of antibodies into the interstitial fluid. These antibodies have the same target as the antibodies on the surface of the sensitized B cells. When stimulated by cytokines from helper T cells, a plasma cell can secrete up to 100 million antibody molecules each hour. Memory B cells perform the same role in antibody-mediated immunity that memory T cells perform in cell-mediated immunity. Memory B cells do not respond to a threat on first exposure. Instead, they remain in reserve to deal with subsequent injuries or infections that involve the same antigens. On subsequent exposure, the memory B cells divide and differentiate into plasma cells that secrete antibodies in massive quantities. A Y-shaped antibody molecule consists of two pairs of polypeptide chains, one pair of heavy chains and one pair of light chains. Each chain contains both constant segments and variable segments. The constant segments of the heavy chains form the base of the antibody molecule. B cells produce only 5 types of constant segments. These are the basis of a classification scheme that identifies antibodies as IgG, IgE, IgD, IgM, or IgA, as we discuss a little bit later in this module. The structure of the constant segments of heavy chains determines the way the antibody is secreted and how it's distributed within the body. For example, antibodies in one class circulate in body fluids, whereas those of another class bind to the membranes of basophils and mast cells. The heavy chain constant segments, which are bound to constant segments of the light chains, also contain binding sites that can activate the complement system. These binding sites are covered when the antibody is secreted, but become exposed when the antibody binds to an antigen. The specificity of an antibody molecule depends on the amino acid sequence of the variable segments of the light and heavy chains. The free tips of the two variable segments form the antigen binding sites of the antibody molecule. These sites can interact with the antigen in the same way that the active site of an enzyme interacts with the substrate molecule. Small differences in the structure of variable segments affect the precise shape of the antigen binding site. These differences make the antibody specific for different antigens. The distinctions are the result of minor genetic variations that occur during the production, division, and differentiation of B cells. A normal adult body contains roughly 10 trillion B cells, which can produce an estimated 100 million types of antibodies, each with a different specificity. An antigen-antibody complex forms when the antibody molecule binds to its corresponding antigen molecule. Once the two molecules are in position, hydrogen bonding and other weak chemical forces lock them together. Antibodies do not bind to the entire antigen. Instead, they bind to specific portions of its exposed surface, regions called antigenic determinant sites or epitopes. The specificity of the binding depends on initially the three-dimensional fit between the variable segments of the antibody molecule and the corresponding antigenic determinant sites. A complete antigen has at least two antigenic determinant sites, one for each of the antigen binding sites on an antibody molecule. Exposure to a complete antigen can lead to B cell sensitization and subsequent immune response. Most environmental antigens have multiple antigenic determinant sites, and entire microorganisms may have thousands. Haptens, or partial antigens, do not ordinarily cause B cell activation and antibody production. Haptens include short peptide change, steroids, and other lipids, and several drugs, including antibiotics such as penicillin. However, haptens may become attached to carrier molecules, forming combinations that can function as complete antigens. In some cases, the carrier contributes to an antigenic determinant site. Antibodies will then attack both the hapten and the carrier molecule. If the carrier molecule is normally present in the tissues, the antibodies may begin attacking and destroying normal cells. This process is the basis for several drug reactions, including allergies to penicillin. Body fluids may contain 5 classes of antibodies, or immunoglobulins, or Igs, or IgG, IgE, IgD, IgM, and IgA. These classes are determined by differences in the structure of the heavy chain constant segments. For this reason, the classes have no effect on the antibody specificity, which is determined by the antigen binding sites. This slide and the next cover the 5 types. So IgG is the largest and most diverse class of antibodies, about 80% of all of them. IgG antibodies are responsible for resistance against many viruses, bacteria, and bacterial toxins. They can cross the placenta, and maternal IgG provides passive immunity to the fetus during embryonic development. Anti-Rh antibodies produced by Rh-negative mothers are also IgG antibodies and produce hemolytic disease of the newborn. IgE attaches as an individual molecule to the exposed surface of the basophil in mast cells. When an antigen is bound by IgE molecules, the cell is stimulated to release histamine and other chemicals that accelerate the inflammation of the immediate area. IgE is also important in the allergic response. IgD is an individual molecule on the surfaces of B cells, where it can bind antigens in the extracellular fluid. This binding can play a role in the sensitization of the B cells involved. IgM is the first class of antibodies secreted after an antigen is encountered. IgM concentration declines as the IgG production accelerates. Plasma membrane cells secrete individual IgM molecules, but it polymerizes and circulates as a 5-antibody starburst. The anti-A and anti-B antibodies responsible for the agglutination of incompatible blood types are IgM antibodies. IgM antibodies may also attack bacteria that are insensitive to IgG. IgA is found primarily in glandular secretions such as mucus, tears, saliva, and semen. They attack pathogens before they can gain access to internal tissues. And IgA antibodies circulate in blood as individual molecules or in pairs. Epithelial cells absorb them from the blood and attach a secretory piece which confers solubility before secreting IgA molecules into the epithelial surface. The formation of an antigen-antibody complex may cause the elimination of the antigen in several ways. First is neutralization. Both viruses and bacterial toxins have specific sites that must bind to target regions on the body cells before they can enter or injure those cells. Antibodies may bind to those sites, making the virus or toxin incapable of attaching itself to a cell. This mechanism is known as neutralization. 2. Precipitation and agglutination. Each antibody molecule has 2 antigen binding sites, and most antigens have many antigenic determinant sites. If individual antigens, such as macromolecules or bacterial cells, are far apart, an antibody molecule will necessarily bind to 2 antigenic sites on the same antigen. However, if antigens are close together, an antibody can bind to antigenic determinant sites on 2 separate antigens. In this way, antibodies can link large numbers of antigens together. The 3-dimensional structure created by such binding is known as an immune complex. When the antigen is a soluble molecule, such as a toxin, this process may create complexes that are too large to remain in solution. The formation of insoluble immune complexes is called precipitation. When the target antigen is on the surface of a cell or virus, the formation of large complexes is called agglutination. For example, the clumping of erythrocytes that must take place when incompatible blood types are mixed is an agglutination reaction. 3 is the activation of the complement system. When an antibody molecule binds to an antigen, portions of the antibody molecule change shape. This change exposes areas that bind complement proteins. The bound complement molecules then activate the complement system, which destroys the antigen, as we discussed previously. 4 is the attraction of phagocytes. Antigens covered with antibodies attract eosinophils, neutrophils, and macrophages. These phagocytize pathogens and destroy foreign or abnormal plasma membranes. 5 is opsonization. A coating of antibodies and complement proteins increases the effectiveness of phagocytosis. This effect is called opsonization. Some bacteria have slick plasma membranes or capsules, but opsonization makes it easier for phagocytes to hold onto their prey before they engulf it. Phagocytes can bind more easily to antibodies and complement proteins than they can to bare surfaces of a pathogen. 6 is the stimulation of inflammation. Antibodies may promote inflammation by stimulating basophils and mast cells. And 7 is the prevention of bacterial and viral adhesion. Antibodies dissolve in saliva, mucus, and perspiration coat epithelia, adding an additional layer of defense. A covering of antibodies makes it difficult for pathogens to attach and penetrate body surfaces. The initial immune response to an antigen is called the primary response. When the antigen appears again, it triggers a more extensive and prolonged secondary response. This response is due to the presence of large numbers of memory cells that are primed for the arrival of the antigen. Primary and secondary responses recur in both cell-mediated and antibody-mediated immunities. So let's look at the pattern of antibody production over time to see if the differences between the primary and secondary responses. The primary response takes time to develop because the antigen must activate the appropriate B cells. These cells must then differentiate into plasma cells. As plasma cells differentiate and begin secreting, the concentration of circulating antibodies makes a gradual, sustained rise. During the primary response, the antibody titer, or the levels of antibodies in the plasma, does not peak until 1 to 2 weeks after the initial exposure. If the individual is no longer exposed to the antigen, the antibody concentration then declines. The antibody titer declines because 1, the plasma cells have higher metabolic rates and survive for only a short time, and 2, suppressor T cells release suppression factors that inhibit the further production of plasma cells. Further suppressor T cell activity does not begin immediately after exposure to the antigen. Also, under normal conditions, helper T cells outnumber suppressor C cells by more than 3 to 1. As a result, many B cells are activated before suppressor T cell activity has a noticeable effect. Activated B cells start dividing immediately. At each cycle of division, some of the daughter cells differentiate into plasma cells, while others continue to divide. Molecules of immunoglobulin M, or IgM, are the first to appear in the bloodstream. The plasma cells that produce IgM differentiate in only a few cycles of B cell division. Levels of immunoglobulin G, or IgG, rise more slowly because the plasma cells responsible differentiate only after repeated cell divisions that also generate large numbers of memory B cells. In general, IgM is less effective as a defense than IgG. However, IgM provides an immediate defense that can fight the infection until massive quantities of IgG can be produced. Unless memory B cells are exposed to the same antigen a second time, they do not differentiate into plasma cells. If and when that exposure occurs, the memory B cells respond right away, faster than the B cells stimulated during the initial exposure. The response is immediate, in part because memory B cells are activated at relatively low antigen concentrations. In addition, these cells synthesize more effective and destructive antibodies. Activated memory B cells divide and differentiate into plasma cells that secrete these antibodies in massive quantities. This secretion is the secondary response to antigen exposure. During the secondary response, antibody concentrations and titers increase more rapidly and reach levels many times higher than they did in the primary response. The secondary response appears even if the second exposure occurs years after the first. The reason is that memory cells may survive for 20 years or more. The primary response develops slowly and does not produce antibodies in massive quantities. For these reasons, it may not prevent an infection the first time a pathogen appears in the body. However, a person who survives the first infection will probably be resistant to that pathogen in the future, thanks to a rapid and overwhelming secondary response. The effectiveness of the secondary response is one of the basic principles behind the use of immunization to prevent disease. So we've now examined the basic cellular structural and chemical actions that follow the appearance of a foreign antigen in the body. In the early stages of infection, neutrophils and NK cells migrate to the threatened area and destroy bacteria. Over time, cytokines draw increasing numbers of phagocytes into the region. Cytotoxic T cells appear as arriving T cells are activated by antigen presentation. Last of all, the population of plasma cells rises as activated B cells differentiate. This rise is followed by a gradual, sustained increase in the activity or the titer of circulating bodies. The basic sequence of events is similar when a viral infection occurs. The initial steps are different. However, because cytotoxic T cells provide an adaptive defense and NK cells provide an innate defense.